It's 4 p.m. here, and I would like to say good morning, good afternoon, and good evening, because I understand we have people attending this webinar from all over the world. I would like to introduce our Priya Banerjee, who's our second speaker in this series of webinars that we have organized since last week. And I hope all of you are comfortable and safe wherever you are. And with that, I would like to say that it's a great pleasure to introduce Priya. I have known him since 2017 when we did an experiment together, actually for the first time when we were trying to understand this phase separating droplets. And this was done together at Rockefeller. And it was that year when Priya started actually his independent group at the University of Buffalo in 2017. And he and his group is interested in understanding how living cells utilize phase transition of scaffolding proteins and RNA molecules to organize their intracellular space into membraneless condensates and control specific signaling functions such as gene regulation. His laboratory is actively developing tools to dissect structure and dynamics these condensates employing high-resolution fluorescence microscopy, optical tweezers, and polymer physics-based modeling. With this, I would like to give the mic to Priya, and I hope we all enjoy this talk a lot, and looking forward to some interesting Q&A. One more thing before we go ahead is that I would prefer if all of you were to use Q&A mode in Zoom for asking questions, just because then it's easier to have a look at it all in one place. We would also try not to have all the questions during the talk, but wait till the end of the talk, unless there is something which you need urgently answered, where you can then raise a hand and ask that question to Priya or to me, and I can answer that during the talk. But otherwise, we can wait till the end, and then we have enough time for a nice Q&A. And yeah, looking forward to some interesting discussions. Priya, the mic is yours.
All right. Can everybody hear me okay, Evan?
I can hear you well.
Okay, perfect. So I'm going to assume that everybody can hear me okay and can see my slides. Thank you very much, Evan and LUMICKS, for organizing this webinar today and the invitation to present some of our recent work on phase separation of protein and nucleic acid assemblies. As one of the defining feature of today's seminar, would be talking about how correlative fluorescence and force microscopy could be utilized to study emergent structure and dynamics of these condensates, which are believed to be present in many cellular organelles. So, due to intense research in the last 5 to 10 years in the field of membraneless organelles, we now know that cells utilize these sets of bodies, right? They are called biomolecular condensates, which are membraneless. They are enriched in protein and nucleic acid. So if you look at the nucleus of an eukaryotic cell, you might see such bodies such as nucleolus, and nuclear speckles, histone bodies, and so on. Nucleolus is one of the most well characterized so far in terms of their structure and function, because it is where ribosome biogenesis takes place. If you go to the cytosol, you also see many prominent membraneless bodies, such as stress granules and processing bodies, which are involved in RNA metabolism. So overall, the idea is basically cells utilize these dynamic bodies which lack membrane-bound structure to perform some sort of active or repressive functions. If you pick inside such organelles, you'll see that these are basically condensed liquid state of proteins or protein-RNA assemblies, and some of them have complex fluid-like properties that goes beyond simple liquid-like behavior. But overall, what you see or think about when you think about these organelles is basically this model of phase transition, where polymers such as protein and nucleic acid come together and create some sort of assemblies which is in dynamic equilibrium with the solution, which is enriched in solvent molecules and depleted of these polymer molecules, right? And these proteins are dynamic within these assemblies. They're moving around, and that contributes heavily towards this liquid-like behavior. Although it's just last 10 years, we have been talking about such bodies and actively looking at their structure and function in biology, we should not forget that phase separation has been predicted by many prominent scientists in the field. So one of the most important paper, I think, that came from Harry Walter and Donald Brooks' group. So they put together this hypothesis almost 25 years ago that phase separation is very common, and that is contributed by macromolecular crowding in a living cell. So in the first part of my talk, I'll focus on this particular aspect of phase separation, how molecular crowding in cellular environment could promote or modulate phase separated structures. If you think about the underlying physics of this process, we can capture many aspects by simple Flory-Huggins theory. So the free energy of a polymer solvent mixture could be expressed as the term mixing entropy right here. So phi is the volume fraction of your polymer, 1 minus phi is the volume fraction of your solvent. And mixing entropy always going to be costly, so it will prevent phase separation because you're going to a more ordered state. So interactions, and this interaction term captures all the mean-field interactions that could be effectively described by this parameter called chi parameter, as Flory introduced in 1940s. It captures the interaction between polymer and the solvent. So green molecule here is your polymer. So polymer interacts with the solvent, the grays are the solvent. You also have polymer and solvent interaction. And overall, the driver of this phase separation could be net polymer interaction, which could be attractive or repulsive polymer solvent interactions. So in the context of today's talk, I'd like to think about these interactions between proteins and RNA molecules which are disordered. And this interaction could be sequence encoded. And there could be interactions that are already well known from the protein science field, such as ionic interactions. So cationic moieties can interact with anionic charges. Also, there could be aromatic residues which can interact via pi-pi interaction. Cation can interact with a pi system and hydrogen bonding interaction. So these interactions are already well studied, but in the context of how proteins or RNA protein molecules can phase separate, we need to revisit these interactions given a set of polymer sequences we have. We should also think about sequence-independent interactions such as crowding, as I mentioned in the previous slide. Which would be an entropic force that might be sequence independent and would be contributed by what you have within the solution other than the polymers that undergoing phase transition. Now, when you talk about interaction, we have to be careful, and at the same time, we have to utilize the wealth of literature that we have already from many, many years of research, that interactions between molecules could be a defining feature of what type of material you form. So for phase transition, you need interactions which are dynamic and of low energy. So that can give you some sort of dynamic bodies which can behave as a liquid. But if you have too much interaction, it can form elastic material which can be reversible, and protein precipitation or crystallization could go in that route. From the perspective of phase transition, this irreversibility could be important because many cellular organelles are known to undergo a process called aging, which is a maturation process where a liquid-like organelle might actually go to a solid-like state. And there are some reports that suggest that this particular process might contribute to neurodegenerative disorders such as ALS and FTD. So one might have to understand first what contributes to a given set of molecules, right, of which undergoing phase transition, what contributes to their liquid-like behavior, and what does it take for this assembly to evolve over time to go from a liquid-like state to a solid-like state, and whether that particular transition is functional or is related to the disease. When you talk about these fluids, right, that are formed by proteins and RNA molecule, we acknowledge that these are associative polymers. So you have a polymer network within these condensates where polymers are interacting with each other and undergoing some sort of collective motions, which could be viewed as rotation. But without going too much into the details of how these fluid dynamics of condensates are governed, one can acknowledge very simply by looking at how protein-protein or protein-RNA interactions are modulated in time and in terms of energies. For example, if you think about this polymer network at a given point, suppose there is this interaction happening, we can easily think about this interaction in terms of the energy. So if you have stronger interaction, the lifetime of this bonding, which is a physical bond, would be higher, suggesting that if we have a collective motion that could be reflected by this type of interaction energies, and if you have attractive interaction of higher orders, you might have slower dynamics, and that could be reflected in diffusion and droplet fusion and so on. Now, these are important features of a condensate because protein-RNA interaction could be encoded by a motif type of interaction, which can be folded motifs, or disordered domains where you have repeat sequences. For example, if you have arginine-rich sequences which are positively charged, or lysine-rich sequences which are positively charged, they can interact with nucleic acids via multivalent electrostatic interaction. That is something I'm going to focus on later part of my talk. So these disordered repeat sequences could give you the license of multivalent interaction, and the energy of that interaction or the strength of such interactions might be important for understanding how the fluidity of these condensates are regulated. Also, such interaction could be changed by external factors such as pH, or mutations in the protein or nucleic acids, or another associated factors present or not present. And that could lead to some sort of regulation or misregulation, so to speak, in diseases of these condensates. So it's important, right, to understand how these interactions collectively shape the fluidity of these condensates. And we need to do that, we need techniques that I'm going to talk to you about in next couple of slides. Okay, so with that, my lab has been working on answering two specific questions for the last couple of years. We would like to know the molecular driving forces that govern the phase separation of these complexes, which are formed by multivalent disordered proteins and RNA. And we would like to know what contributes to the regulation of structure and dynamics, and whether we can relate, right, how they phase separate with the structure and dynamics of this condensed phase. One of the most important technique, or most commonly used technique in the field to study diffusion in a condensed phase is fluorescence recovery after photobleaching. So what you do here in this cartoon, you could see, suppose these are all single protein molecules, you bleach it by an intense laser. So the molecules undergo some sort of photo deactivation permanently, but if there is diffusion, molecules from the surrounding medium could diffuse to the bleaching region, and you'll get some sort of rate of diffusion. So here I have a simulated movie that shows that if you bleach it, and if there is high internal dynamics or diffusion, you could actually regain the loss of fluorescence, and that is what you have for sorry, I lost the PowerPoint by playing that movie. Just give me a sec to, there are several movies and I'm hoping that I'll be able to play all those movies. So hold on. Sorry about that. Let's have to go back to where I was. Yeah, so I think we are back. Okay. So I think we're back now, And again, I apologize as I played the movie and wanted to get out of the movie screen. I could actually see the PowerPoint crashed. Okay, so using Frap again, we could actually study how protein molecules or RNA molecules diffuse within these condensates, and that gives you some sort of idea about the liquidity. Although strictly speaking, there could be diffusion behavior, those for molecules with differential interactions with the condensate matrix, and that could give you some sort of false positive. So you need techniques beyond just Frap. What we have done also using the correlative fluorescence microscopy, which LUMICKS offer, we could utilize a very simple assay where you trap using optical tweezers, you trap two protein or protein-RNA droplets, and then you, under controlled environment, you could actually see them undergoing fusion, and that particular experiment gives you two very important properties of condensates or any fluid, which are surface tension and the viscosity, okay? And I'll tell you why trap-induced fusion is better than just random fusion of droplet that you might see in many papers. And we also have done those kind of studies, but the resolution is limited by video microscopy speed. With optical tweezers, what we are doing here, we are not analyzing the video microscopy, although you can do that, but we are looking at the force relaxation as trap 1 is sort of handing over the droplet to trap 2 during fusion, and the force or the laser signal from trap 1 undergoes some sort of relaxation. And that is a very high resolution because you can record that at 12 microsecond time scale resolution, which gives you at least 2 orders of magnitude higher resolution, temporal resolution for doing fusion analysis. Here I'd like to show you a couple of videos which I'll refer to later. So for example, if you have highly fluid-like droplets, again, I'm trying to play this video and I hope you can see it and my PowerPoint doesn't crash. Yeah, so what's happening here, if you have highly fluid droplets, they undergo fusion very nicely using optical trap. And here we have some sort of aged droplets which are harder, and you see that they resist fusion because they have undergone a transition which is from fluid to gel-like. And as we are trying to push them together, we will not be able to do it. So using this correlative force fluorescence microscopy, what we could do is we could implement a workflow where we have these flow cells that's again provided by LUMICKS. So you don't have to go anywhere, everything is provided by them. You just have to do the experiments. So here we are flowing all the condensates, and you could see, we have 2 optical traps that could trap these, and then once you're satisfied with their sizes, you could turn on the fluorescence and they will undergo fusion. So with this simple Frap fusion assay, one can interrogate how nanoscale diffusivity dynamics is regulated by a set of factors that could be mutation, that could be environmental factors such as molecular crowding or pH. At the same time, you can also do droplet coalescence by optical trap, and you get very high resolution readout of micron-scale fluid dynamics. Okay, so our model system was a protein called fusin sarcoma, and this is a multifunctional RNA-binding protein. The relevance of this to study the phase behavior or aggregation of this protein comes from its biological features, such as the protein is localized in one of the major membrane-less organelles, cytoplasmic stress granule, and the protein undergoes aggregation in ALS, which could be contributing to the pathobiology related to this particular protein misbehavior. From a protein scientist perspective, if you look at this protein's domain architecture, you have 2 different domains. One particular domain is called prion-like domain, which is largely devoid of charges. It has polar residues such as serine and glutamine and glycine, and tyrosines as aromatic residues, and these are repeats of, not perfect repeats, but there are repeats of those. So it's a low complexity prion-like sequence. On the C-terminal part of the protein, you have these RNA binding domains where you have this RNA recognition motif that is folded. But there are several arginine glycine-rich repeats which are unfolded and contribute to interactions with RNA via multivalent repeat sequence-based interactions. Okay, so we wanted to study several features for today's talk of the phase separation of this protein. But for today's talk, I'll focus on one specific idea: how macromolecular crowding inside a cell can change how proteins phase separate, or what are the behavior of the phase separated condensates. So to study that in a controlled environment, we looked at multiple different polymers that can mimic crowding. And the data that I'm going to show you is on PEG 8000, but we have used at least 8 different crowders, which shows similar behavior for this protein. So what we did here first, we took the 2 hubs of the protein, the prime domain, which has very different sequences than the RNA binding motif, and we show that both these domains can phase separate, and the phase separation is enhanced by macromolecular crowding created by PEG. So what you're seeing here is some sort of a state diagram which suggests what the condition is for phase separation. So if you have 150 micromolar protein, it phase separates without any crowder. But if you increase crowder to 100 mg/ml, the protein phase separates heavily at 30 micromolar. Okay, so the green dots are phase separated state, and the gray dots are no, or gray triangles are no phase separation. For the RNA binding domain, we see similar trend with PEG, suggesting that macromolecular crowding heavily facilitates phase separation. And if you're wondering what is the PEG concentration that might correspond to crowding in a cell, that is up there 150 and onwards. Okay, so we have just mild crowding condition, and we see a heavy impact on phase separation of these 2 domains. And it also tells you something: the protein has overlapping mechanism of phase separation, because both domains are capable of undergoing phase separation on their own. And so it's of no surprise that when you combine these 2 domains and create the wild type protein, the protein phase separates at very low concentration. So without any crowd, the protein undergoes phase separation in 2 micromolar, but as you increase crowding, you could see that the phase separation of this protein is now facilitated, and it's happening at low micromolar concentration, less than 1 micromolar indeed. Okay, and again, where this crowding condition, which is not too high, very much within the limit of what cells offer in terms of crowd. Okay. One of the things I think was interesting to us is to study what happens to these condensates when we change crowding, right? For example, if you start with 2 micromolar protein, it forms droplets at all these crowding concentrations. So the question is, does anything change when you change the crowding? And that could be important, because if cellular crowding levels are changed, that might change how protein droplets behave inside a living cell. But again, there is no study prior to this. So we looked at how PEG changes the droplet properties. So first we did our diffusion measurement using Fraps. And I have 2 movies here, one with 150 mg/ml PEG. So I'm gonna bleach one of these droplets and see if you see how they recover. So let's play the higher crowding first. Okay. And you could actually see, I hope you can see that we created a bleach region, and it's very slowly recovering. It doesn't completely recover even after 5 minutes. Now, if I go to 50 mg/ml, which is relatively lower crowding, and you'll see that within a blink of your eye, that the diffusion will take place, and you may actually miss the whole bleaching experiment. So let's try that. And I hope you saw that everything recovered very fast, suggesting that just by changing crowding, your phase separate condensates appear to be the same under confocal microscope, but the dynamics of proteins within this droplet has changed. We can do better. We can quantify this, and using a 2D model, which suggests, again, a relative change in apparent diffusion coefficient by 4-fold, from almost 16 microns square per second to 4 microns per second. And in the mobile fraction, which suggests how much, what is the fraction of molecules mobile within the liquid condensates, it changes from 100% to 40%, okay, suggesting by changing this much PEG, you're changing the physical properties of these condensates. When we did our fusion experiments with optical trap, we even saw dramatic changes. For example, if you go back one slide, if we are looking at just the black and the red curve, there are not much changes here. But if you look at the fusion dynamics with 0 mix per ml PEG and 25 mix per ml PEG, we see that the fusion of these droplets almost an order of magnitude slowed down by addition of this much crowding agent. And if you increase the PEG concentration to what's relevant in terms of cellular crowding, the droplets undergo hardening. The video I already showed you when I introduced the technique, and they do not undergo fusion at all. You keep trying to fuse them and they just stick and they don't fuse, suggesting that macromolecular crowding is not only changing their condensation behavior, but also physical properties. So with this set of data, we conclude that correlative force fluorescence microscopy could be very effective in capturing dynamics of these condensates spanning nano to microscale. And utilizing that, we could quantify how crowding not only can promote phase separation of this protein foods, but also changes the material properties, which might be of importance because many condensates are changed during aging, as I mentioned earlier. And a model emerges from this study that crowding changes the state of condensation of the ribonucleoprotein from nothing to liquid to viscoelastic gel. And this could be very important and interesting if you think about how crowding could be controlled in cells. So this study came right after we published our paper. They looked at how ribosome concentration, which they used as a crowder in living cells, cytoplasm, could tune crowding. And if you tune the crowding by ribosome, you could also tune phase separation versus no phase separation, okay? Our study is suggesting it's not just about phase separation, it's about the condensed physical properties, which can also be tuned. So that is very subtle, and that might be important to understand how cells might change their functionality by changing fluidity. So that's something we're very interested in to follow up, and some work are in progress. Okay, so I'll move on to my next part of the talk. I introduce this protein FUS, which is RNA-binding protein. I think obviously one would ask this question, so what happens to this protein when it interacts with RNA? So the next part of my talk will be focusing on this specific feature where the protein undergoes condensation with RNA, or RNA regulates how the protein phase separates. Cutting a long story short, I'll give you the result immediately. So RNA has a dual role here. What we have done over the past few years, not only us, but others in the field, including Alberti's lab in Germany. So we found that FUS can form droplets at a very low concentration of RNA. So if you take, say, 5 micromolar of FUS, the protein does not undergo any phase separation under these conditions. But if you add very little amount of RNA seems to promote phase separation. And if you take, say, 30 micromolar of FUS, that undergoes phase separation without any partner. But again, if you add a little bit excess RNA, the droplets disappear, suggesting that RNA inhibits phase separation of this protein. So what this set of data suggesting is basically a model where RNA auto-regulates phase separation of this RNP. And again, I'm going to focus on FUS today, but this is a defining feature of many nuclear RNA-binding proteins, TDP43, HNRNPA1, and so on. So the model is quite general when we talk about the role of RNA in this context. Okay, so we could quantify this phase behavior and show that there is this window of composition where phase separation is promoted. So here we are plotting concentration as a function of this ratio of RNA to protein, and we're using some homopolymeric RNA. You can do cellular RNA. It doesn't really matter. We have tried many different RNAs. The data suggests here that with poly-U RNA of different length, you get this behavior which we call reentrant, which tells you that you start with no phase separation, and then you add RNA, which takes you to phase separated regime, and then you add a little bit more RNA, that the system re-enters to a homogeneous mixture. And that's why the name reentrant phase transition. And if you do microscopy, you will see that the phase separated droplets are only observed where you have this composition we call regime 2. Region 3 is highly interesting, where you have higher amount of RNA and that dissolves condensates. To understand the mechanistic behavior here, we wanted a model system, a simplified model system. So we looked at the part of the protein which binds to RNA and has this multivalent arginine glycine rich motif. The reason we looked at the arginine glycine rich motif is because the RG repeats are not only present in foods, but many eukaryotic RNA binding proteins, such as other H and RNPs. In fact, 50% of eukaryotic RNA binding proteins include this RG-RGG repeat sequences. So that's from the functional perspective. From the pathological perspective, we have these highly toxic polypeptides, which are glycine arginine repeats and proline arginine repeats, and they are linked to this hexanucleotide repeat disorder in ALS and FTD. So we would like to understand how these repeat arginine sequences contribute to phase separation, and what is the mechanism when they interact with RNA. Okay, so we started off with this simple polypeptide from the third RGG box of FUS, and arginines are highlighted here. So you could see that they are well distributed, and again, they are repeated, and there are other sequences as well. So we could recapitulate the reentrant behavior with poly-U RNA and RGG. You see this rise and fall of light scattering intensity. So you have this window of phase separation given by this composition. We can utilize that and create simple diagrams, which we call thermodynamic state diagram, that tells you within this two component concentration plane, what are the composition that leads to phase separation. So what you have here, this color intensity that is basically proportional to the scattering intensity, and the dotted line right here gives you the coexistence line. So basically, within the dotted line, the system undergoes phase separation. If you are in regime 1 versus regime 3, there is no phase separation at all. And the propensity of the system to phase separate is shown here using the color scheme, and color scheme corresponds to the scattering intensity. The bottom line is we can actually reconstitute a system from the full-length protein which undergoes reinfant condensation with RNA, and we can study its thermodynamic properties. To understand the mechanism here, we first thought about what are the interactions that you can see between this arginine-rich domain and the RNA. So Ibrahim, a graduate student in the lab, he started asking this question in a slightly different way. He was asking if there is a sequence code that might be at play here. Now, he made some observations. For example, the RG-RGG motif, where you have these arginines, which are positively charged, they do not contain another positively charged residue, which is lysine. So if it is all about the positive charge of arginine and negative charge of RNA, it doesn't make sense because there is no lysine. So there is something specific about this high bias towards arginine. So he looked at these two specifically designed constructs, RG-RGG5 and KG-KGG5. So these are pentameric construct of this pentapeptide, so 25 residues each, and you have arginines selectively mutated to lysines. And what we are going to do is we're going to look at their phase behavior with RNA to ask this question: if there is a sequence code, and if that sequence code is beyond just simple electrostatic model. And again, this study is published last year. When we got that state diagram, we see that the arginine-rich domain or arginine-rich peptide shows a higher two-phase region, a larger two-phase region than lysine, and you could also see in the turbidity plot. So lysines have narrower regime of phase separation than arginine, suggesting that there is a sequence code. How about RNA? Can we change RNA? So we change RNA from poly-U to poly-A RNA. So poly-U has this homopolymers of one-membered RNA base, which is uracil. We changed it to adenine, which is two pi-membered rings. And we see that the widening is even more significant for arginine than lysine here. So again, green is arginine, red is lysine. And what we have seen that at higher concentration, which is not too high, 2.5 mg per mil peptide, the reentrant nature of the condensation disappears because you simply don't have enough RNA to add to make the droplet dissolve, suggesting maybe that you're also changing the properties of these condensates. So what we have done then to understand this phase behavior in terms of sequences, we put together this proposal that we have to look beyond just electrostatics, and we found from the literature that there are excellent reports suggesting that lysine and arginine can interact with the RNA bases beyond electrostatics. For example, cation pi and pi-pi interaction. And since arginine has this guanidinium group, it is better poised to do cation pi than lysine, which doesn't have that kind of pi system. So it's a combination of the short range cation pi pi interaction beyond electrostatics that might be tuning how this condensation behavior is governed, right? And a rank order emerges from this comparison from all these papers that I cite here. It suggests that arginine adenine would be stickiest, and then lysine uracil would be the least of the interactive partners here. So we propose that this widening of the two-phase regime is due to short-range interactions encoded by protein and RNA sequence. And that tells us that the arginines are stickier than lysine, and we'd like to know if that changes the condensate dynamics. So what we have done after this, of course, we had this assay in place, the correlative force fluorescence microscopy and fusion Fraps assay. So we did that with these 4 combinations of peptide and RNA condensates. So this is basically, again, summarizing the techniques we are using. And we have seen a very dramatic behavior when we change protein and RNA. For example, if you start with, say, RGG5, and if you have poly-A RNA, they undergo fusion under optical trap at 350 milliseconds. But if you take arginine with poly-U, it's just 60 milliseconds, which is well less or way fast compared to what you have with poly-A RNA. Changing arginine to lysine, you look at this lysine with poly-A, it undergoes fusion at 2.8 millisecond timescale, which is almost 2 orders of magnitude slower than with arginine and poly-A. Okay, so we can quantify this data as a scale relaxation time, and we see that overall arginines are taking longer time to fuse than lysine, which is expected based on the interactions present within these chains. And then we also see that poly-A RNA has a longer fusion relaxation time than poly-U RNA. Again, 2 pi system versus 1 pi system. That's what we predicted based on existing literature. We then looked at the diffusion properties, and we see exactly the same rank order. For example, if you look at this green profile, which is lysine with poly-U, everything recovers within 2 minutes to 100% level, versus if you have arginine with poly-A, there is almost no recovery, as if this is a gel-like condensate. Okay, suggesting again, and the other 2 are in the middle, suggesting again the rank order that we predicted based on the interactions work well at the nanoscale, where we look at diffusion, as well as at the micron scale, where we look at droplet fusion. But one thing I would like to point out here, and the importance of doing these measurements not only at one length scale, such as Fraps, but also to fusion, because if you just look at the profile black line here, which is arginine and poly-A, the profile suggests that there is almost no recovery, suggesting it is a gel-like condensate, which may not be true if you look at its fusion profile. The droplets undergoing fusion at 350 millisecond timescale, which is pretty fast if you think about 2 JLO droplets undergoing fusion. They may not undergo fusion, as I showed you for fused droplets. So you see that there are impacts of interaction changing from arginine to lysine, but the motions in micron scale timescale versus nanoscale timescale could be independent, and they could be somewhat decoupled. So it's important to do correlative measurements to get more information than just doing a simple measurement. We went back to our full-length fused protein and asked the same questions, whether interactions are governed by the sequences. So these previous slides were focused on this arginine versus lysine on synthetic polypeptides. Now at fused, we did not change arginine to lysine, we changed poly-A versus poly-U RNA. And although fused is a promiscuous binder of RNA, you could see that if you just take the arginine-rich LCD, which is a 280 residue polypeptide, you see similar behavior, which is shown right here, almost 1 order of magnitude slower fusion if you have poly-RNA versus poly-uRNA, and the third or GG box of FUS behaves similarly as well. We can look at diffusion as well, and we see a twofold change when we change RNA. Again, this twofold change is distinct from tenfold change in the fusion, suggesting the macromolecular motions at different length scale could be decoupled. Okay, with this set of data, we conclude that RNA can auto-regulate phase separation of ribonucleoproteins such as FUS, and interactions beyond simple electrostatics such as cation-pi interaction or pi-pi interaction could complicate how reentrant dissolution takes place. So with simple model system, we could show that cation-pi and pi-pi interaction oppose the dissolution of this condensate, and it also regulate the condensate dynamics across nano to microscale. In the last part of my talk, I'll be trying to tell you a brand new story about how the structure of these condensates are regulated. Now, we spend a lot of time thinking about their dynamics, but I would like to focus on structure as well, because one specific thing here that this two-phase region is quite large, so you can actually move around in terms of composition, and the question is whether you will be changing their properties such as structure. Now, in the cells, these membrane-less organelles that I pointed to earlier, they're not simple droplets. They are structures which are best described by subcompartments. For example, in nucleolus, you have these three subcompartments: granular component, dense fibrillar component, and fibrillar component. In stress granules, you could also see a core and shell kind of structure. And this is the part of the most interesting. So if you overexpress just a simple RNA-binding protein TDP43, you could actually see that the protein itself from condensates, which are multiphasic, meaning they have core shell structures, but the inside structures are of low density than the outside structure. Okay, so all these different types of droplets in cells suggest that there could be more complex structures other than just simple droplets. So the question is, does this can this be recapitulated in a simple model system such as these protein RNA mixtures? So what we have done here first, this is done almost 3 years ago. So we tried to first see if we can recapitulate structures by flowing one of the components such as RNA, because the condensates dissolve when you have more RNA, right? So we wanted to study without dissolving them, if I change their composition, how does this affect the structure of these condensates? Okay, so we are going to actually travel across this white line. So using a simple mixing device, what you can do is you can jump from low RNA to high RNA. So first, we allow our condensates to grow a little bit on the surface. And then what you could do is you could flow RNA and you could start seeing this core shell-like structure forming. Some of these structures are only stable for a few minutes. And strictly speaking, that is due to the non-equilibrium nature of these structures. So we thought this is highly interesting and deserves further studies. So we spent last 2 years studying these structures and to ask a very simple question: are these can these be stabilized under conditions which are not non-equilibrium, and study their properties? What kind of structures are we forming here? Because they resemble some of the structures visualized in cells. What we have found that, yes, indeed, we can form them by simple mixing RNA and protein. So here, what we are doing here is we are mixing this arginine-rich polypeptide with RNA. So the polypeptide concentration is 4.4 mg per ml. And if I increase the RNA concentration, as you can see in the top row right here, initially you form droplets, they grow, but then they stop growing, and they form these well-dispersed condensates. And then at some point, the condensates become extremely dynamic, look like fuzzy droplets. And something magical happens when you add more RNA. And again, these are all mixed in a test tube before imaging, and we image them for hours, and you see these hollow condensates emerging. You can study their properties again using the confocal microscopy part of CTRAP, and you could actually see very nice ring-like structures. So you have RNA and protein enriched within the ring. and the center appears to be hollow. You can also study diffusion properties. Now, Frap may not work because the diffusion is extremely fast in the dilute phase. So we did fluorescence correlation spectroscopy, and again, this is done using the CTRAP from LUMICKS. So it has excellent resolution in terms of photon sensitivity. And we could study this by simply positioning our confocal volume within these condensates versus outside or within the rim. And you could see that if you are looking at the rim, the diffusion is, the half time of diffusion is in milliseconds versus when you have, when you are looking at condensate inside, that we call it lumen versus outside, they're almost 3 orders of magnitude faster, okay? And the half time is in microseconds now, suggesting that the inside part of these hollow condensates are similar in density than compared to outside, and they are indeed a dilute phase, okay? All right. We can also do liquid-like property measurements such as Fraps. So on the left, you have Frap, and we are frapping on the rim, and you could actually see that we frapped and it went away. We can also trap these condensates, which are hollow, using optical trap and undergo fusion. So here you could see that they undergo fusion very well. They first form a multi-compartment condensate and that collapses. You can also trap a droplet and a hollow condensate and try to see if they're undergoing fusion, and you could actually see that they do. So using optical trap, you could study these properties very easily. We can also go back and forth in terms of RNA. So again, this is done using LUMICKS microfluidic cells, and you can see that we have 2 droplets, and we're gonna flow RNA under very controlled environment to make this transition to see if we can create hollow condensates from spherical droplets by RNA flow. So you could see that we have these 2 condensates, RNA are being flown, and using bright field microscopy, you see a very nice transition of these condensates going to hollow condensate structure, and this is under optical trapping. You can also take away RNA and see how these hollow condensates react. So we did that using RNAs, and you could see that this is made way faster. This total span is 15 minutes or so. The condensate, actually the internal lumen collapses over time, it shrinks, and then at some point it will be extremely unstable and collapses, suggesting RNA degradation by RNAs would create a reverse transition from hollow condensate to condensates. Okay, all right, so now what we have is basically to study if this is specific to the protein system we are studying, or is it related to or can we actually see these for a series of systems? And we have seen that systems which undergo phase separation via predominant heterotropic interactions could undergo phase separation into hollow condensates. So it's a quite remarkable generic feature of these systems. And finally, to understand the mechanism, we have studied their properties utilizing molecular dynamic simulation. We utilized a statistical mechanical based approach, which was introduced by Sklopsky. So under compositionally discrete mixtures, what you have that you only have partially condensed chains, which we call tadpoles, and as the tadpoles form, they can come together and form micellar condensates, which are highly stable because these empty or naked chains are on the surface. And at certain concentration of these empty chains or micelles, you can start forming vesicles. So with our collaborators at Iowa State University, David and Morley, they could implement this model in their MD simulation, and you could see a tadpolar structure where you have a condensed head and a free RNA tail. And then you have micellar structures under excess RNA condition where the condensates are decorated by excess RNA on the surface. And if you keep pushing the overall concentration of the system higher and higher, you see a transition from micelle to vesicle, where now RNA has larger volume to be distributed because it has outside and inside, which are accessible to the RNA chains, which are not condensed. Okay. All right, so again, this particular study is available for you to read. If you're interested to dig in more, it's available in bioRxiv. We conclude from this part that protein-RNA complexes can not only form condensates which are membrane-less, but they can create vesicle-like structures which are lipid-free. And that happens due to associative phase separation of the polymer. I didn't show you data, which also part of this manuscript, that these vesicular assemblies, these hollow condensates, have properties which are classical for lipid vesicles. For example, local ordering, such as liquid crystalline structures, size-dependent permeability, and selective encapsulation capacity. Okay, so with that, I'd like to thank the people who contributed greatly towards the overall growth in last few years in our lab. As I've been mentioned, we only started 3 years ago, but we have two very talented postdocs, Richu and Mahadi, and very talented graduate students, Ibrahim and Charanpit, and some very talented undergraduate, Hannah and Andrew. And our collaborators are UB and beyond, and our funding agencies, NIH at UB, and above all, LUMICKS for their continuous support. So this is our group within the C-Trap room, and you could actually see this is the C-Trap. So if you haven't seen C-Trap in person, this is how the instrument looks like. Very sophisticated, looks like a spaceship to me, and we utilize it pretty much day in and day out, and they have extremely well customer support. They will take care of you. With that, I'd like to stop here and thank you for joining. And I'm not sure, since I can't see you, I'm not sure if you got all the movies right, or all the slides went well to you, but I'll be happy to answer any question you may have. Thank you very much.
Thank you, Priya. I think that was a wonderful talk. I was particularly stoked by the last part, which was very, very interesting, but I guess we can talk about it later. And yeah, discuss the aspects of how you did those experiments.
Yeah,
but
thank you.
For now, yeah, I would like to thank you again. It was a wonderful talk. And like you said, we are always happy to help you. Your experiments are always something that motivates us to do better at LUMICKS. So there are already a few questions in the Q&A section. I think we can go to them very soon. What I can do is I can read out to you the questions, and then you can answer them. And you can also read the questions in the Q&A section if you miss something.
Okay, fantastic. Let's do that.
So let's start with the first question. We have a question from Bede Potts. He's interested in the transition from rough-looking condensates at 11 milligrams per milliliter RNA to hollow condensates at 22 milligrams per milliliter. Do you think that the RNA itself is condensing at that concentration and recruiting the protein to the surface?
So we do not have any evidence under these conditions that poly-U phase separates under, again, solution conditions. So last year we published a study itself studying how poly-U phase separates, and it needs Crowder and it needs high magnesium concentration or sodium concentration. So under these conditions that we are studying protein-RNA condensation, RNA does not phase separate in our experiment. So the answer to your question, I guess no, we have characterized it really well so far, that RNA is not condensing at this concentration. It's rather protein-RNA droplets and RNA is on the surface.
I hope that was clear to you, Bede. If not, you can already type in again, and then we can come back to it after the next few questions. Yeah, the next one we can go to, and I might be pronouncing your names wrong, so please apologize. I apologize already. So we have a question from Vasilis. Would you predict similar types of condensates between poly-RG, poly-KG, and DNA molecules?
So by similar type, it's very vague. So one of the things I think is important to understand that not all types of RNA would give you liquid-like condensates. So it might be a very interesting question to understand what happens if you go beyond certain types of RNA. So far we have been focusing on this poly-U RNA, poly-A RNA. We studied cellular RNA, and we see quite heterogeneous structures. So for example, if you go to poly-A RNA, and again, that's published in the JACS paper, we see viscoelastic gels. And with some of the mRNAs, we also have seen, solid-like structures. So the overall differences between arginine and lysine would be the same. Okay, so arginine would condense our DNA more than lysines. And that's probably due to the way it interacts with DNA, right? And that's something we're studying right now. But we do not know whether the condensate structure will be similar, because I don't think we'll be forming liquid-like condensates if we use very rigid DNA molecules.
Again, same. If you need a follow-up, please write it down in the Q&A. There is Pilong, who likes your talk a lot, and he's asking, for the vacuole-like structures, is there a diameter-dependent trend?
So these vacuoles like structures that we have seen, they're very polydispersed. So when you say diameter-dependent trend, I'm not very clear what trend we are talking about, but what we have seen in that movie where we are creating vacuoles by RNA flow, that's very heterogeneous, and that's strictly speaking non-equilibrium. So things are more complicated there. But when we mix two things together and form these vesicle-like structures, we have seen a high degree of polydispersity. Okay, so we have seen smaller droplets or smaller hollow condensates versus larger. So when you say diameter-dependent trend, I'm not sure what trend again we are talking about. So please feel free to add a comment so that I can better understand you.
We can go to the next question. Maybe first, Daniel, thanks you for the very interesting talk. Coming to Sankar Sundaram, he again thanks you for the nice talk. He's wondering if the reentrant phase system, in the reentrant phase system, is it possible only for the condensate to shrink, the hollow, but the RNA remains homogeneous?
It's a possibility. I do not have a good answer for your question. Yes, it's a very exciting possibility. But using molecular dynamic simulation, you could actually recapitulate some of those videos that I shown. And that's also part of our bioarchive paper. Please go and take a look. But our understanding so far is RNA is not homogeneous. RNA has excess RNA conditions. Under excess RNA conditions, if you are forming micellar condensates, RNA is on the surface of those condensates. And if you're forming vesicular condensates, again, they are very close to the surface. And we're talking about both surfaces, the internal surface, which is the lumen, and the external surface. So RNA may not be homogeneous, but it's a good possibility that you mentioned. And again, we have to probably do more studies, experiment and simulation to answer your question.
There's one more question. It's from Shengqi Xiang. Is the surface and the interior of the droplet the same? Is the droplet homogeneous?
We haven't done experiments to tell you that, but we see under micellar conditions, where you have RNA on the surface is different than the internal part of the droplet. So we do see that under disproportional mixture composition, the interior and the surface of these droplets are not the same. And we have studied that using MD as well as some microscopy techniques to clearly show that the excess RNA is on the surface, and that changes things quite a bit.
Thanks. We have one more question from Hemashri. Can you elaborate on how to choose molecular crowders for nucleic acid and protein interaction? Rather, what are the criteria to select the molecular crowders?
So that's a very, very nice question, and I do not have a good answer, because when we started looking into the same problem, I did not have any clue. So we took crowders which are widely utilized, such as PEG, dextran, and FICAL, and you might see crowder specific effects, because what you see sometimes that even we claim that the crowders are very inert, they are not. They might actually introduce some sort of interactions with the DNA, RNA, and protein chain, and that could be system specific too. So again, I do not have any answer, any specific answer to your question, but what we have done to sort of circumvent that problem, we studied quite a few crowders rather than just one crowder, and everybody in the crowding field probably has their favorite crowders. So I'm not a crowder person, We just published one study using Crowder, so we tried to stay away from choosing a Crowder because we just don't have enough information to tell you, okay, this Crowder is the best Crowder, because cells have different types of Crowder than what we use for macromolecular crowding studies. So perhaps there are studies where they used instant cytoplasm, like instant coffee. So instant cytoplasm could also be used as a Crowder. So we probably should test more Crowders. That's the answer. That's what I think.
Thanks. There's another question, a follow-up from Sankar. Additionally, do you know if structured RNA versus unstructured, so long homopolymers such as poly A, influences reentrant properties of condensates?
Yes, so I think so, because what we have done with poly U versus poly A and other RNA to show that interactions are adding up. For example, if you have structured RNA where there are specific interactions that you cannot have for homopolymers such as poly A, that will contribute greatly to the reentrant properties of the condensates. Okay, so it's a collective phenomena where you have interactions such as short range interaction. And when I talk about short range, I only mentioned about cation pi and pi pi, but you have cation pi pi pi, van der Waals interaction, sequence specific interactions, structural interactions. All of these interactions going to be important to think about how reentrant transitions are influenced. So the answer is absolutely yes. The structured RNA would change things quite a bit, and there might be a possibility to see a generalized behavior if you study, or if you end up studying a large number of sequences, which is what we're trying to do right now. Yes, sorry.
There's a question from Soumya Bothra. She says, in the thermodynamic state diagrams, what was the third kind of interactions?
I think we're talking about three different phases here. So the regime 1, 2, 3. So regime 1 and regime 3 are homogeneous mixtures, which are basically protein-RNA complexes that are soluble. Regime 3 was a soluble complex that you form with high amount of RNA when you mix it with protein. Okay, so so it's not the type of interaction; it's the type of complex. It's the type of complex you form when you have excess RNA compared to the protein.
Thanks. Yeah, we have a few more questions. I really like the energy of people asking questions, and they are really interesting as well. So we have a question from Miriam, who's asking, as the hollow condensates are energetically unstable, is there a way to stabilize them? And a follow-up question is, under which conditions are those hollow condensates relevant in vivo? And can they be filled with something, for example?
Yeah, so to answer your first question is yes, they can be stabilized if you think about cross-linking type approaches, which could be used to stabilize them. We can also engineer protein or RNA sequences that could lead to a little bit more stable interactions. So yeah, that's a possibility. Part 2 of your question is, under in vivo conditions, we already have seen TDP43 forming these condensates. So I think we're talking about, so from our study, we can only talk about excess RNA conditions. And, there is a science paper from Simon Alberti's group where they measured the concentration of RNA within the nucleus, and the concentrations are not very far from what we have used in our experiments. So under those conditions, you could actually see that these hollow condensates are forming with many RNA-binding proteins. And we expect to be very generic. And they can be filled with something, yes. So, the part that I didn't really talk about, maybe you can go and take a look at our bioarchive paper. We could selectively encapsulate single-stranded DNA, single-stranded RNA, single-stranded DNA, double-stranded DNA, and protein molecules. So they can be filled up with things, yes, absolutely. And in cells, the TDP43 condensate, at least I know that it was filled with nucleoplasm. So it was a nucleoplasm filled space. So that's, that could, give you a different kind of membrane-less condensate than just simple, isotropic liquid droplets.
Fantastic. There's one more question from Jingwen Shen. Part of it is similar to what you answered before. So are these hollow condensates visualized in vivo, like in cells or organisms? And then
Yeah.
whether the hollow transition needs specific type of RNA or any RNA is fine to induce this.
So we have done with cellular RNA. So we took cellular mixture of cellular RNAs from yeast, and if we mix it with RG-rich polypeptides, we see this condensate. So I think it's quite a generic feature. Again, we haven't done enough to tell you exactly what I think about specific RNA structure versus sequence. Now going back to the cells, we only have limited cellular data so far, but it's we already have seen that TDP43 is one of the RNA-binding protein that can do it. And there are other examples from Drosophila, membrane organelles. So there is this protein called Oscar. It forms germ granules that have hollow structures, and that is dependent on RNA binding as well. So what I think here is it could be a defining feature for many protein or RNA system or systems that undergo phase transition by this heterotopic interactions. And I think those could be relevant for organizing things inside a cell. But again, we have very limited information from cellular data yet, but still they are indeed visible like TDP43 and Oscar condensates.
One more question from Dieter. In your condensate coalescence experiment, does the velocity with which the condensates are approached affect the relaxation time?
Yes, so it doesn't really affect the relaxation time. It mess up completely with your experiment. So you have to do in a very controlled way where you see the handoff. So it's a competition between when two droplets are approaching together, right? It's a competition between the trap trying to keep the droplet and the condensates are undergoing fusion due to coalescence, the surface tension and viscosity, right? So to get a good readout, you need to figure out or optimize the system in a way that this handoff is really happening, so that you can see the laser, the trap on laser signal is decaying as this handoff is taking place. And of course, you do that not just standalone, you also do video. So you are recording video microscopy. So for slower system, you could utilize that data to figure out whether your trap data is telling you exactly the same thing or not, and then utilize the trap data after the validation for faster fusion experiments. So yes, so there's quite a bit of optimization that's needed to be
done. Thanks. There is a question from Ashish. He's asking, is it possible to differentiate condensates and hollow condensates on the basis of turbidity assays? And can we relate this transition with development and functioning of nucleolus?
The first question is, the answer to the first question is no. I mean, they are still going to give you turbidity, so I'm not sure how one would do that. So my take would be no. And regarding the development and function of nucleolus, I think I'm not in a position from this data to tell you that there is some sort of consequences and how these hollow condensates are forming and how they are functioning. But that's a fascinating topic to look into, I guess.
Thanks. There is Pinaki who liked your talk a lot, and thanks you for it. And he's asking, for the reentrant phase transition with increasing RNA concentration, is it due to screening of electrostatic interaction regime 3? Since cation pi interaction competes with this phenomena, can we predict phase separation tendency from the sequence of aromatic and charged residues?
Yes, so there is of course screening of electrostatic interactions in regime 3, but you can also think about how the complexes are overcharged because of non-stoichiometry nature. So you start with, so if you do a light scattering measurement which can measure the overall charges of the species, you will see a charge inversion. And that is because when you have little RNA, the complexes are positively charged. And when you have a lot of RNA, the complexes are negatively charged. So you see a very nice electrostatic transition from positively charged complex to negatively charged complex. And I think one of the idea that we proposed in these papers on reentrant phase transition, that electrostatic repulsion plays a big role. Now this repulsion is actually counteracted by cation pi types of interactions. Now going back to your question about if we can predict the tendency of phase separation from the sequence of aromatic and charged residues, absolutely, why not? But I don't know how to do that. So somebody else have to do it, and I need to just use that prediction and do experiments.
Thanks. There's a question from Hilong again, who's asking, is the vacuole-like structure likely to form for very small diameters, like tens or hundreds of nanometer?
Yes, so we have seen, like I said, we have seen quite a bit of heterogeneity or polydispersity, and we have seen, even sub-micron condensates could have hollow structures. Yes, but again, we cannot go beyond 250 nanometer using normal confocal microscopy or 300 nanometer because of the resolution issues.
There's a question from Sina. Is there in vivo evidence? So we answered that, I think, earlier. And do you compare it with the core and shell structures of stress granules? But those have solid cores, as far as I understand. Do you think the molecular mechanisms are similar to stress granules?
No, yeah, we think when we talk about nucleolus, when we talk about stress granules, I think there are more things going on with hollow condensation. I think hollow condensation is something different. When you think about those structures, I think multiphasic condensates are more valuable so far to understand how coexisting liquid droplets can give rise to a core shell structure. So you have a gel-like condensate within a fluid-like condensate. The structure that we describe here, the hollow condensates, they're like lipid vesicles. They are not your core shell structure that you think about for droplet inside a droplet morphology. So the molecular mechanisms in terms of these particular phenomena could be very deep. And again, we are only looking at a tRNA system where you have one protein, one RNA system. But for droplet inside a droplet morphology, you need at least one more component. So suggesting that the molecular mechanisms could be different, and it could be a collective phenomena coming from the multi-component nature of the system.
We have a couple of questions left. So one is from Jingwen again. The hollow condensate phenotype is very striking. Whether these hollow transited condensates play a role in biology, we, I think, answered that. Unless you want to elaborate any more.
No, I again want to tell you that we know so little about this. These are the structures we just saw, and our paper is under review right now, and we hope to find out if there are any biological functions.
Yeah, Soumya, thank you for your answer before. But there's one more question which is somewhat similar, but are the physical characteristics important biologically or relevant?
Yeah, so absolutely. Again, I don't want to make too strong of a connection here because we haven't studied the biological relevance. But the idea is this, right? So if you have a condensate that you think fluidity affects function, what we are suggesting here is that we can tune the fluidity by simple physical rules, which are protein-protein interaction, protein-RNA interaction. So, and we can characterize that. So if you have a mutation in a given protein that leads to, say, solid liquid to solid phase transition, would be in a better position to look into that in detail and characterize the mechanism. And then if you want to target that using drug-like molecules, I think we might be able to do that and test everything in cells, right? So I think they are highly relevant in biology, but I don't want to make too strong of a connection here because, again, we haven't studied that connection directly.
Thanks. Pilong says that many of their protein-only systems have formation of vacuole-like structures.
Okay, so I'll be intrigued to discuss more offline. So if you would like to discuss this more, you can send me an email. I hope you have my email, or I can. I think you will be able to find my email. So I'd be happy to learn more if you want to discuss it offline.
Pilong, I can also send you email, Priya's email, if that's okay with you, Priya.
Yeah, that will be fine. Thank you.
Yeah. So there's a question from Priyanka who's asking what happens to hollow structures upon aging.
We haven't studied aging directly. We have only done experiments for like few hours to study their whether they disappear, and they do not disappear. So I think that's a fascinating question, but we do not have much data to tell you what happens if you keep them for an elongated period of time. And that goes well, I think, with the theme that some of these condensates undergo some sort of glassy transition or aging transition. So yeah.
All right, thank you. There's another question. Sorry, I think I sent. Yeah, from Vasilis again thanks us, and he liked your talk a lot, and also the discussion. There's a question from Charlotte. If you add fuse to RNA instead of adding RNA to fuse, do you think you would form the same type of hollow structure, or is there something special about the interaction between fuse at the edge of the droplet, example surface tension with RNA?
So again, I'd like to clarify that the structure that we have shown, the hollow structure, they are not with full-length fuse. Full-length fuse is a different entity altogether because it's not just simple homopolymeric type of protein. Okay, it has diblock nature because of its prion domain. So the question about fuse RNA, we do see structures which are hollow structures, but what is striking about these RG domains and RNA basically that they do not have this diblock nature or amphiphilic nature of polymers that undergo phase transition into hollow condensates. Now, let me answer your question. What we think happening here is there is some sort of steady state, right? It's still a metastable state because it's phase transition. But what we see here that you can mix these two together and you form these droplets. So I don't think that it will be specific to what you add first and then the other, but you might see differences. And again, without doing the experiment, I cannot really dig into any deeper than what we have so far. So overall, we think these are steady state structures. So if you mix them together, what we have done for the hollow condensates in this specific work, we see that they form very nicely. So it's basically you mix these two, and by the time you take a sip in your coffee, they are there. So, but when you are forming it under non-equilibrium condition, it could be order of addition dependent. So again, we have to do the experiment to think about it.
I would like to thank all of you for attending this talk, and most importantly, I would like to thank Priya for taking us through this wonderful journey, and we are looking forward to more of this interesting work in the future. I would also quickly like to share my screen, and so I would like to also invite you to our webinar next week. It's Dr. Raya Sorkin at the Tel Aviv University, who will be talking about membrane remodeling in life processes and looking at membrane interaction one vesicle at a time. It would again be a very interesting talk, and I welcome you all to come for it. And lastly, and lastly, if you have any more questions, you can direct them to me at my email address, which is a.ramaya@lumicks.com, or you can write email to info@lumicks.com, and we will make sure that these questions reach Priya, and we have the answers for you as soon as possible. So yeah, thank you again, and have a great day and be safe.
All right, thank you, Evan.